Hello everyone, I'm here to discuss some introductory dilutions that you're going to be using in a microbiology lab. Generally in microbiology, we use dilutions that are done at a ratio of 1 part of our sample and 9 parts of sterile diluent or buffer, giving a total volume of 10 parts. So this 1 to 9 part ratio is very, very common and used in almost all calculations that we do. It can be varied as long as this ratio is maintained. We can have other ratios that are done at 1 part sample and 99 part of buffer or volume. Now this would give us a 1 in 100 dilution. Generally, we write these numbers in scientific notation. So a 1 in 10 dilution, 1 part sample, 9 parts buffer, would be written as 10 to the minus 1. A 1 in 100 dilution, 1 part sample and 99 parts buffer, would be written as 10 to the minus 2. A general formula that you can follow is the volume of your sample divided by the total volume. This includes your diluent volume and your sample volume. Using larger sample sizes is actually a very common practice in the lab. 1 milliliter or 1 gram of sample is a small portion, and it can't always represent the entirety of the product that you're testing, a food or any other sample. So government methods commonly say to use 25 or even larger volumes, 25 grams of sample or more. In our labs at Centennial, we will use commonly 10 grams. This is a happy in between. There's enough sample size that we can get a good representation of the whole sample, but we're not going to use an excessive amount of media, supplies, and reagents, etc. So using different volumes, we're going to keep the same ratio of that 1 in 10 dilution. So 25 grams would be in a total of 10 times that volume, 250 grams. The example shown with the picture here is 10 mils of sample into a total of 100 milliliters of volume. So you can see 10 mils of the beaker there comes from the sample. The other 90 mils comes from a sterile buffer solution. So you have 10 mils in a total of 100. When you reduce that fraction, it still is a 1 in 10 dilution. Other dilutions can be used as long as that ratio is maintained. So 25 grams would be placed into a total of 250 grams. Reducing that fraction, you're still going to get a 1 in 10 dilution, or a 10 to the minus 1. Using 50 grams, we would put 50 grams into a total of 500 grams, 10 times more than the sample volume. And that would be reduced down to a 1 in 10 fraction, or 10 to the minus 1. When you're doing these types of questions, you have to remember that the bottom number of your fraction, so 250 or 500 in these two examples, is the total volume. So if I'm to ask you how much buffer you would need, you have to subtract the original sample size. So 25 grams in a total of 250. That 250 grams is made up of 25 mils of sample, and the rest of that is your buffer volume. So you would need to do a very simple calculation of 250 minus my original 25 grams of sample, and that would equal the 225 mils of buffer that you need to add to this sample. So this is what it would look like if you're combining these different dilutions to do a microbiological calculation. You would have initial culture, so in the pink jar there, 3.5 times 10 to the 9 bacterial cells in every 1 milliliter of that solution. You're going to take 1 milliliter of that solution and transfer it into a tube with 9 mils of sterile buffer. 1 mil into 9 mils. This gives us a 1 in 10 dilution. We're going to do the same thing again with the next tube. You're going to take 1 mil out of the tube that you just created and transfer it into another 9 mils. This is going to dilute your sample yet again. So we can see here, if you follow the yellow line across, the dilution in the first tube is 1 part sample, 9 parts of buffer. In the second tube, those are going to be combined. So we're actually going to have only 1 part of the 1 milliliter transferred into that second tube that is representative of the original sample, and the other 9 parts of the 1 mil transferred is the buffer from the first tube. So we're actually increasing the dilution every tube we go along. You can see on the white line on there, the expected cell count. Our original cell count is 3.5 times 10 to the 9 cells in every 1 milliliter. In our first tube, we've diluted it 10 times, one tenfold dilution. So now our cell count is 3.5 times 10 to the 8. In our second tube, the cell count is 3.5 times 10 to the 7. We've reduced it by another tenfold dilution each tube as we go down the line. Now we can see the math here to how we calculate the total dilutions. The first tube we did a 1 in 10 dilution. The second tube we've also done a 1 in 10 dilution. When we multiply these two fractions, 1 times 1 in the denominator side is still a 1, and 10 times 10 in the numerator side is a 100. So now our second tube is going to have a 1 in 100, or 10 to the minus 2 dilution. In our third tube, we're actually going to have 1 in 100 from our second tube that we're going to dilute another 10 times. So this is now going to become 10 to the minus 3, which is equivalent to 1 in 1,000 dilutions. So we're going to continue on, and these dilutions will compound and add to each other. We have two things to be careful on in terms of terminology. We have a dilution ratio versus a dilution fraction. A dilution factor. And these are both used differently when you're calculating your raw data to work back to the original concentration of bacteria in your sample. A dilution ratio is always written with a negative exponent. So it's actually a very small number, 1/10, 0.1, or 1 in 100th, 10 to the minus 2, which should be 0.01. A dilution factor is a large number. So this is 10 to the positive exponent, 10 to the positive 1, and it would get larger and larger. Now these are used differently when we're working with raw data in our calculations. If we have performed an experiment and diluted our sample, making 10 tubes where we did a 1 in 10 dilution 6 times over and over, and we had a bacterial count of 139. We now have to work backwards. There wasn't 139 cells in every 1 milliliter of our sample because we diluted it 6 times before we actually put it on the plate to grow these colonies. So this can be calculated in two different ways. We can take our 139 colonies and divide it by the dilution ratio. So we have 139 divided by 10 to the minus 6. So this is your dilution ratio, which always has the negative exponent. That I definitely need a calculator to do, and I cannot do in my head. The other option for calculating these things is to use a dilution factor. So this would be the equation where you take the cell count on the plate and you multiply it times the dilution factor. So 139 cells times 10 to the positive 6. This I can do in my head. It's very easy. I'm just going to round up my 139 and maybe move my decimal over so that I get a nice number presented in scientific notation. So the point here is that you're going to divide by a dilution ratio, and you're going to multiply your cell count by a dilution factor. I hope this gives you a good introduction and a basic foundation to continue on with this section on microbiological calculations. Thank you.